Looking at all these young faces, I feel very good. Second, I am awed because you've got distinguished scientists, eminent scholars here, and I admire what they have accomplished. I have seen what Rameshji has done, and I have seen some of these other scientists who are here and their accomplishments. But at the same time, many of these wonderful scientists, and they are seen as seers and sages as we think about scientists. In their labs, isolated, but today, as I heard them, eloquent, very, very insightful, and I sat in some sessions, and I'm very, very pleased to see that we have got a crop of people's stars rising who are going to be able to do a whole lot for mankind. And today, as the session began, and as from the Holy Quran, you said a verse, and then you talked about it, and that resonated with me, that those who work for humanity, those who serve others, not for themselves, that they are the ones who are at the present time pillars, foundation for the future. So as I began, the second reason that all these people, eminent scientists, that they are able to present their thoughts cohesively, coherently, and at the same time with a great deal of insightful. And third, I looked at this huge book and hundreds of papers. And many times, sitting in the United States, I think about India. Obviously, all these people who are from here do think back about India. And in my own discipline in international law, I'm very, very pleased to tell you that I come back, I do pro bono teaching at the Indian Law Institute. I'm the honorary professor now at the University of Delhi, Faculty of Law. And these coming generation of international lawyers gives me a great deal of hope. And seeing all of you who are going to be there tomorrow as scientists and eminent scholars, teachers, I want you to know that it's wonderful to be a good teacher and wonderful to be a teacher, because it's my students, when they talked about the Vedananda Center for International Law, it's my students who created it. One of them put $1 million in a few months, others matched it. Couple more millions from former students has come in. Very small, about $4 million center. And other students raised about $1.5 million, and there is a professorship and a chair in my name. What else can you ask as a teacher? So to all my friends who are teachers, I've got to tell you, be good to your students. And I think the point simply is that usually as an academic, you die or you retire. And then perhaps the school, perhaps the university might have a chair in your name. But this center and this chair, it's not in memoriam. I'm still there, standing, and moving around and teaching. And so I think I want you to know that I'm very, very pleased to be here, and I'm so glad that you permitted me to crash this party.
Thank you so much, sir, for your enthusiastic words, your inspiring and motivational words. You have made time for our event. Besides your tight schedule, we are really honored and humbled. Thank you so much. We are also privileged to welcome Dr. Thomas M.S. Chang, Founder President, International Society for Nanomedical Sciences, as our chief guest for today. He invented and primed the first artificial cell back in 1957 by using nanotechnology and nanobiotechnology to prepare artificial cells containing biologically active material with 10 to 20 nanometer membrane thickness polymers, cross-linked proteins, and other membranes. Dr. Chang is also the Director, Artificial Cells and Organs Research Center. He is rightly called the father of artificial cells. Honored to have you here, sir. May I please request you to kindly address the August gathering.
This is truly an interdisciplinary symposium. We have an international lawyer here. They just give us an excellent talk. And we must remember, nanoscience is an area that we do need lawyer, social, ethical people to come and give us feedback so that we don't go the wrong direction, especially it's moving so fast. Okay, having said that now, I would like to represent the International Society of Nanomedical Science. Actually, I'm just the honorary president. I prefer younger people to be the founding president. In fact, we founded the society, but I refuse to be the president. And he and me and also Professor Best, the secretarial, want to thank Professor Sachuan and Professor Hasner for having together organized such an important meeting with such distinguished guests coming to support his meeting. And all the fellow researchers and scientists in the audience, it's a pleasure to come and talk a bit. Professor Sandra asked me to talk a bit this morning about artificial cell, and so that I don't have to talk too much in this lecture. So the thing is that I must apologize this morning. I just want you to be more exciting. So I put in a lot of animation. But unfortunately, animation is difficult when you go from my computer to another computer. So I spent my lunch. Remember, I did it for you. I didn't have lunch. So I move out everything except very simple thing. Next slide, please. Oh yeah, that's fine. During intermission, I have a number of young students come and ask me, how did you do artificial cell when you're still a student? Since we want to show young people what to do, I told them that I didn't start when I was a BSc. You don't need a lab. You don't need any money to practice how to invent things. I told them that when I was just one in primary school, high school, I can always think about how to solve problems. And in fact, you should all start, even you are master or younger, even high school. For example, when people were thinking about planes are being very big, what to do with the runway. We have to make it very long, make the brake, and and all that. So I start thinking, and I said, why don't we put a parachute at the end of the plane? When the plane lands, we open the parachute. People thought I was crazy, but now people do use it. So remember, you can think and invent without any money, without any lab space, and as I go along, I'll show you the example. And number two, you would like to go out of the box. Don't be confined by the box. Don't be confined by tradition in order to go ahead. For example, you see, artificial cell was originally trying to make red blood cell. But you don't want to be confined by biology. You want to be able to add vaccine, compartment, magnetic thing into the thing in order to enlarge the area. And don't worry, because scientific journal, they want you to do that because I'm so surprised science, nature accept this type of crazy idea. So next slide, next slide, please. So since then, with international group each one with their own brain pushing down this area. This is expanded tremendously internationally. Next slide, please. For instance, I showed you this morning, so I'm going to talk about it. You have many, many different configurations around the world. Next slide. And these are all examples of what people around the world doing, new idea, new approaches. As a result, nanomedicine is advancing so fast. The more people, the better. Next slide, please. So because of that, there are many areas of application. I don't even list half of it, but just some example. Next slide, please. Now, just to show you one example of nanomedicine, this area. Next slide, please. Next slide, please. The number of submissions increased so rapidly, and that the publisher has to increase the volume. And I want to say this again for the people, especially graduate students. They cannot afford to publish that many papers. They cannot afford to look after all this, so they start to do it by open access and charge a fee. I don't like it, but if I don't, they would have to go bankrupt. So I bargained with them. They agreed for me to have 25% of the paper without any charge. And I said, okay, I would do this by asking people to send me the paper. I look at it, especially from academic world, only from academic world. If it's good, then I'll give them a code. They can then submit it. But don't submit it until I give you a code. Otherwise, they will charge you. Okay, next slide, please. So now, I'm going to go over briefly blood substitute. Next slide, please. So there are different ways to do blood substitute. If you follow the tradition, you want a membrane around red blood cell because cell has a membrane. Even nano cell, you need a membrane. But you have to think outside the tradition. You can use nanotechnology. And that's how nanotechnology, nanobiotechnology starts in the corner. Next slide, please. For instance, nanobiotechnology is the assembling of biological material, and this is what happened. Cross-link it to make it more secure, become soluble polymer. Next slide, please. And this was then developed. At that time, nobody was interested, but then it was developed because of HIV, and now it's approved by South America to prevent HIV infection. So the original aim was reached. Next slide, please. Next slide, please. Sorry, I have to keep asking because it goes. Please go back one. Can you go back one? Okay. So other important areas are blood loss, like hemorrhagic shock, like stroke, heart attack, that should be investigated. Next slide, please. But these need more complex system. You can't just use a hemoglobin complex. So what we did very recently, 2015, a PhD student of mine from Tsinghua University from China, we then combined all the enzyme of red blood cell together with hemoglobin to prepare a nanobiotherapeutic. And the next slide, please. Now, since you're doing it artificially, you can put in any proportion of enzyme. Because hemoglobin is the large majority, enzyme is only a small fraction. You can put it up to 6 times concentration of red blood cell enzyme, resulting in 6 times effectiveness of red blood cell. Next slide, please. So this is what happened. So in summary then, we did all the basic research in the laboratory, showing all the problem, and now the next step is to develop this for preclinical study, eventually leading to human use. Next slide, please. I was talking about hemorrhagic shock, but you can also use it in heart attack, in stroke. Why? Because in heart attack and stroke, many cases it's because of the blocking of the artery. Now, since since red blood cells are particles quite big, they cannot pass through the obstruction. As a result, you have stroke and also heart attack. Now, next slide, please. If you now use the solution is not a particle, it can much more easily pass through the obstruction. Furthermore, you have 6 times the enzyme concentration to remove the oxygen radical and the CO2. And this, next slide, please. Now, this is another example of what you can do by just thinking without even doing any research. Next slide, please. Back when I was still just finishing my PhD, I used a slide rule. I don't know how many people know slide rule. I don't think Professor Sandra knows. It has to be very old to know how to use a slide rule. You use a slide rule, and then one day I sit down and calculate. So nanotechnology includes nano-thickness membrane. So I sit down with a slide rule and did some calculation, comparing a big hemodialysis machine with 5 cc of artificial cell with nano-thin membrane. I don't want to go into detail, but over there you can show that it's 100 times. 5 cc artificial cell is 100 times more transport capacity. Next slide, please. So, but then 5 cc doesn't remove too much. You can put anything inside the artificial cell, adsorbent enzyme to convert what goes in. So then you can make a very small device, as you see there. HP means hemoperfusion device. That's 70 grams compared to a whole artificial kidney machine. Next slide, please. This is what happened. I have to do this myself personally in patient because the other doctors wouldn't want to do it because of liability. So since I prepared myself, tested very carefully, I thought I can trust it. So